Hello everyone, welcome to Pathomad. Today I would like to tell you about a case of basal cell carcinoma regarding its microscopic findings. Firstly, this is the clinical image showing you the lesion over the tip of the nose. See, basal cell carcinomas most commonly occur over the face and it is more famously called the rodent's ulcer. The rodent's ulcer because it is prone to spread fastly in and around the face. And this specimen, this lesion was excised and was sent to histopath department in a formalin container. It was measuring 1.5 into 1.5 into 0.8 centimeters. Externally, it is gray-brown in color and the surface is nodular. So we have cut this specimen into two halves like this, and the cut section shows you within in each cut section it is gray-white in color and the two halves were divided into four bits, like each one divided into four bits. That means, so this is the lesion we have cut into two halves. In each half, four bits were given. Four bits were given. 1, 2, 3, 4, 1, 2, 3, 4. Total 8 bits were given for microscopic examination. So this is the low power view. In this low power view, you can see the dermis, appreciate the epidermis, sorry. This is the epidermis. The epidermis invaginating into the dermis is called rete. This is the rete. So this entire thing is the dermis. From here to here, everything is the dermis. Within the dermis, you can find there are certain groups of cells, like in a nested pattern. This is one nest, a group of cells. This is another nest. Many such nested nests of cells are seen. And from the epidermis, the basal layer is invaginating into the dermis to form these groups, to form this nest of cells. And in this slide, you can appreciate this nest. There is a cleft in between this nest and the adjacent stroma. See, this space is there. Space is there around the nest. It is separating this nest from the adjacent stroma, which is called the retraction artifacts. The clefts are called retraction artifacts, which is basically during the process of fixation, the mucin present in the stroma gets dissolved. It gives rise to formation of this cleft between the nest of cells and to its adjacent stroma, which is characteristic of a basal cell carcinoma. And within each nest also, I can find the like cribriform pattern. There are gaps within it, like filter sieve pattern can be appreciated. This is a sieve pattern within one nest. For example, this is one nest cribriform pattern, sieve-like pattern can be appreciated. In the low power view itself, this is again under low power view showing you many such nests are there, and there is a cleft retraction artifact between the nest and the stroma, and within the nest also you can find a cribriform pattern. This is again one such low power view, and here and there in focal area you can find the keratin pearl is also seen. And here, for example, if I keenly observe this one nest, I can find the rim is very like highly differentiated. Like the cells are very disciplined, sitting beside one another, like in a row. This is called palisading of cells. Like they are oriented well vertically adjacent to one another, well oriented cells to one another. That is palisading I can appreciate when you zoom in. See, I can find this typical palisading here. Again, to zoom in at that area, see you can find these cells are palisading. They are adjacent, closely packed, adjacent to one another. Whereas the cells in the center, they are like arranged in a haphazard pattern. In the center, the cells are haphazard, but at the periphery, they are palisading. They are closely packed and oriented in one particular direction. That is called peripheral palisading. Peripheral palisading is very characteristic of nodular BCC, nodular basal cell carcinoma. See, within one such nest, there is peripheral palisading, and within the nest, you can find appreciate sieve-like pattern, sieve-like pattern. which is the cribriform nest. It is characteristic of adenoid variant of nodular BCC. Adenoid variant. This is another nest showing you the cribriform pattern, cribriform nest. Another low power view showing you focusing this keratin pearl. Keratin pearl very focal, very few keratin pearls could be found in this slide. When you zoom in, within the keratin pearl, a nuclei like random nuclei can be appreciated. This is called parakeratotic pearls. Parakeratotic pearls. When you zoom in, see this is the keratin pearl. Keratin pearl. You can find this nuclei. There are nuclei within the keratin pearl. So the nuclei within the keratin pearl, it favors malignancy. It favors malignancy, and hence the characteristic features like the cribriform pattern within the nest and presence of retraction artifacts, nested pattern, all this favor adenoid variant of basal cell carcinoma. Thank you all and have a nice day.